The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us on today's webinar. You've logged on to the Nelson Labs webinar, An Introduction to Evaluating Persistent or Residual Antimicrobial Efficacy of Topically Applied Products in a Clinical Trial Study. Now, just a few housekeeping items before we turn the time over to Chris. If you missed one of our webinars or would like to simply refer back to one, you can always find them on the Nelson Labs website on the On-Demand Webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on Twitter or LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from our introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to achieve patient outcomes and minimize client risk. Let's get started. Today, we're joined by Christopher Beausoleil. Christopher has worked at Nelson Labs Bozeman, formerly known as Bioscience Laboratories, Inc., since 1994 and has been involved in all phases of in vitro and clinical testing of test articles. He has over 27 years of experience evaluating the efficacy and safety of test articles using various standardized test methods and customized protocols for regulatory submissions or to support marketing claims. We do encourage your questions, and please submit them at any time in the questions box. Chris will try to answer as many as he can in the last 10 to 15 minutes of the webinar. Now, beginning today's presentation, I'd like to turn the time over to Christopher.
Thank you, Mike. Hope everyone's having a great day, and I'm looking forward to presenting to you this information. Start off, topical antimicrobial products are an important tool for safeguarding global health. They help disinfect the hands of healthcare personnel, surgeons, and food handlers. They disinfect the skin prior to surgery, injection, or catheterization. And they help to limit the spread of germs and disease in hospitals, healthcare facilities, food service industry, schools, home, and in the public. First, I'd like to define some important terms that we use when we do our evaluations. The first is immediate antimicrobial effectiveness. This is a quantitative measurement of both the mechanical removal of a microorganism by washing and the product's ability to rapidly inactivate microorganisms residing on or contaminating the skin surface. Persistent antimicrobial effectiveness is the quantitative measurement of a product's ability to prevent microbial recolonization of the skin surfaces, either by microbial inhibition or lethality after application of the test product. In this example here, you have the skin with microorganisms present on the skin. You treat it with an antimicrobial. That reduction of the microorganisms on the skin immediately after application is your immediate antimicrobial efficacy. Then after a period of time, that recolonization is prevented. If it's prevented by the antimicrobial, we call that a persistent antimicrobial efficacy. The third effectiveness is the residual antimicrobial effectiveness. This is the measurement of a product's cumulative antimicrobial properties after it has been used repeatedly over time. That is, as the product is used repeatedly, it is absorbed into the stratum corneum of the skin and as a result prevents microorganism recolonization of the skin surfaces. It's a little different from the other ones in that you have the skin and then you do multiple applications with the antimicrobial. You have a contaminating microorganism, in this example here, a transient microorganism. After a period of time, that contaminating microorganism is reduced. So we call that a residual antimicrobial efficacy. Other terms that we use are resident microbial skin flora. These are the microorganisms that survive and multiply on the skin, forming a stable population. A transient microbial skin flora, these are microorganisms that contaminate the skin but do not form a stable population. In all our studies, we use neutralizers, inactivators, or neutralizing additives in our sampling fluids. These are a chemical or physical means of inactivating or quenching the activity of an antimicrobial agent at the time of sampling. So at immediate sample time, you want to neutralize that antimicrobial during the sampling process so that you don't have a continuing killing effect in the sampling fluids during the assay procedures. And we validate these neutralizers using ASTM E1054. There are two procedures or sampling methods that we use to sample microorganisms from the skin. This first one is the cup scrub procedure or cylinder sampling procedure, or we call it either in all our protocols or publications. Sterile cylinder is held firmly onto the test site. This could be a glass cylinder, or we use a stainless steel cylinder. We find those are easier to grip and hold on to the skin sites. You add a volume of sampling fluid with neutralizers into the cylinder, and then you scrub the area inside the cylinder using a sterile glass rod, a rubber policeman, or a Teflon rod for about a minute. You remove the sampling fluid and transfer that to a sterile tube, and then you repeat that procedure with a fresh volume of sampling fluid for another minute. And then you pull those sampling fluids and assay those to determine the number of microorganisms that resided on the skin. Another procedure that we use to sample microbial flora from the skin, actually uses from the hands, is the glove juice procedure. This, you place loose-fitting, unlined, powder-free gloves onto the hands. You can use a low bioburn bags, may also be used. You add a sampling fluid into the glove with neutralizing additives. You secure the gloves above the wrists, and then you uniformly massage all surfaces of the hands for 1 minute. Now, when conducting a clinical trial study, you need to follow good clinical practices. This is an international ethical and scientific quality standard for clinical trials with principles consistent with the Declaration of Helsinki. ICH E6R2 is the guideline, international guideline for good clinical practice. This describes the content of the study for the study protocol, adverse drug reaction reporting, and general statistical considerations such as design, sample size, and methods of analysis that you need to take into consideration when performing a clinical trial study. All our clinical trial studies undergo an are reviewed by an independent review board or independent ethics committee, an IRB or an IEC. They review the study, and their main concern is the study participants and safeguarding their safety, well-being, and confidentiality. We follow the Federal Regulation 21 CFR Part 56 for institutional review boards. All subjects must sign and must provide informed consent. This is obtained from every subject prior to participation, informing them of the study procedures, expectations, possible risks, potential benefits, and other relevant information to allow for them to make an informed decision whether or not to participate in the study. And we follow 21 CFR Part 50, protection of human subjects, for our informed consents. We also follow good laboratory practices. We have the clinical portion where we take samples from the skin, but we follow good laboratory practice on how we handle those samples once we take them from the skin. Our assaying procedures are an in vitro method, and we follow 21 CFR Part 58, good laboratory practices for non-clinical laboratory studies. Most of the methods that we use for evaluating persistent residual efficacy are under ASTM. There isn't a European method that we also have used for evaluating surgical hand disinfection. So ASTM E1115, this is the standard test method for evaluating surgical hand scrub formulations. And then the European method is the EN 12791. ASTM E1173, that's for evaluating preoperative, precatheterization, or preinjection skin preparations. E1174 is for evaluating healthcare personal hand wash formulations, and E2755 is for evaluating healthcare personal hand rub formulations, or waterless hand sanitizers, hand rubs, those types of products. And E2752 is a standard guide for evaluating residual effectiveness antibacterial personal cleansing products, and E3058 is another residual kill activity of hand antiseptic formulations. I'll go through each and every one of these methods, just give you a brief overview of those so you see how they evaluate the immediate, persistent, residual efficacy. E1115 for surgical hand scrub formulations. This determines the immediate, persistent, residual antimicrobial effects of the surgical hand scrub or rub product. This uses the glove juice sampling procedure. Subjects are refrained from using antimicrobials for at least a week. We recommend 2 weeks, just to allow for stabilization of the microbial flora on the hands. Subjects will undergo a baseline week where 3 estimates of a microbial population residing on the hands will be performed. These are usually done on Monday, Wednesday, and Friday of a test week. Samples are collected following a cleansing wash, or a wash with a cleansing wash formulation. It's non-antimicrobial. We just want to get an idea or get an estimate of the normal flora on the hands, not any transients they may pick up on their way to the lab for testing. Subjects are admitted into the test week or the following week with baseline counts of greater than or equal to 1 × 10⁵ CFUs per hand. So they have to have a somewhat high bacterial count on their hands so that we can detect, we're trying to detect efficacy. So we need to be able to have a baseline to detect whether or not there's a reduction of that population that's on the hands. And this is estimated after the first and second baseline collections. That Friday collection is usually being counted while we're doing testing the next week. During the test week, this is performed either with just a single test day or multiple days. For the single test day, this is where the immediate and persistent antimicrobial effect is evaluated. Subjects will perform a single surgical scrub or surgical hand rub, and then their hands will be sampled at 2 post-scrub rub sample times. Typically, we've done immediate and 6-hour post-scrub times. Over the years, we've done immediate 3 hours and 6 hours. So we'd randomly assigned one of those sample times to a hand, and then another sample time to the other hand. So subjects could be assigned to have their right hand sampled immediately and their left hand sampled 6 hours, or if we're doing a 3-hour sample time, one hand 3 hours, 1 hour, other hand 6 hours. For the 3-hour and 6-hour sample time points, the hands are gloved and secured for the duration of that 3-hour or 6-hour time point. The evaluation of the cumulative effect with an 11 additional scrubs over a 5-day period is how the residual antimicrobial effect of a product is determined in this study. So after that first scrub on that first day and after that final sample, subjects will perform an additional scrub or rub and then return the next day. They'll return each of the next 4 days. On days 2, 3, and 4, they'll perform 3 additional scrubs or rubs with an hour between, or days 2, 3, and 4 with an hour. They'll perform 3 additional scrubs with an hour interval between each treatment. And then on that final day, day 5, which is typically a Friday, they'll perform an additional scrub rub and repeat the post-scrub sampling times that they performed on that first day. Any kind of residual effect would be an increase in reduction over what you saw on that first test day. Now, EN 12791, this is a European method for surgical hand disinfection. This determines the immediate and persistent antimicrobial effects of a surgical hand scrub or rub product. This one, the subjects are required to refrain from using antimicrobials for at least 10 days. It's required that you test 23 to 28 subjects. It's a crossover design, so each subject's going to use both a test product and a reference control product. If you have other products also to use, you'd use a Latin square design if you have more than one test product. The order of use is randomly assigned. Pre and post-treatment samples are taken the same day. Sampling is performed for this. It's using fingertips by rubbing the fingertips of a hand for 1 minute in a petri dish containing TSB without neutralizers. That's for the pre-treatment. And then for the post-treatment samples, they'll do the same kind of rubbing of their fingertips in a petri dish with TSB with neutralizers for the post-treatment sample. Pre-treatment samples are taken following a cleansing wash from both hands. Subjects will rub hands after that sampling. Subjects will rub both hands together and allow their hands to air dry. After that air dry, subjects will then use either the test or the reference control product, whichever one they were assigned to use for the first round of testing. Following that product use, one hand will be sampled immediately by rubbing the finger pads into the petri dish. The other hand will be gloved for 3 hours. So similar to the ASTM method, the other hand is gloved, except this is for 3 hours. And then after 3 hours, that hand is then again sampled by rubbing the fingertips into a petri dish. Subjects then return a week later and then perform that exact same evaluation again with the other product. So if they use the test product first, they'll use the reference product, or if they use the reference product first, they'll use the test product second. The mean reduction for the immediate effect, so that immediate sample, and the 3 hour sustained effect, that persistence of the product, shall not be inferior to that achieved by the reference product. Now for ASTM E1173, this evaluates preoperative, precatheterization, preinjection skin preps. Skin preps that are intended to be used on patients prior to surgery, prior to catheterization, prior to injection. This determines the immediate and persistent antimicrobial effects of those types of products. The cup scrub procedure is used for this evaluation. Subjects refrain from using antimicrobials for at least 2 weeks prior to the test day. Subjects refrain from showering or bathing at least 2 to 3 days prior to test day. We want the bacterial populations on those skin sites to be as high as possible for those sites. We want that high just so that we can actually detect reductions from them. And it helps to simulate worst case scenario that healthcare personnel could be encounter with a patient that comes in for any types of these procedures. Baseline samples are taken from one site within an anatomical region, and the populations are recommended to be sufficiently large enough to detect significant reductions following treatment. Post-treatment samples are taken following that treatment. Any sites that are sampled hours after application, we cover those sites with semi-occlusive dressing. It's just a semi-occlusive catheter dressing with a sterile gauze pad underneath it to cover the site and prevent that site from being recontaminated during that time period. We just want to evaluate the microorganisms that are on that site. The treatment sites that we typically evaluate for these studies are the inguine, the abdomen, the clavicular region, and the median cubital region of the arm. Within each one of these sites, we typically use about 4 sites, one for baseline, and then you have 3 of the other sites for post-treatment sample times. For some of like the abdomen site and the clavicular region, you can add a 5th site to the center of that area if you want. The median cubital region and the inguine region are limited to those 4 sites just by the size of the area. To evaluate preoperative skin preparation, to evaluate a preoperative skin preparation, use a moist and dry skin site. There's an inguine, that would be the inguine and the abdomen. Recommended sample times for this evaluation are 10 minutes, 30 minutes, and 6 hours, but other times may be selected. Immediate efficacy is determined within 10 minutes. We've evaluated other times of less than 10 minutes of like 30 seconds or immediately after treatment. Persistent efficacy is determined at that 6 hour time point. Vascular precatheterization preparations. This skin sites that are areas that may be catheterization sites. These are sites that will be catheterized for long-term admittance for some patients in the ICU or just in the hospital. So they have to have products that are not only effective immediately, but do have some kind of a persistent effect over time to prevent any kind of infection of developing at those sites. Moist and dry skin sites are evaluated. Again, as the inguinal, the clavicular region, and the median cubital region. The recommended sample times for these are immediate 12 hours and 24 hours post-treatment. Other times may be selected. We've done these evaluations 4 days and 7 days after treatment. Again, immediate efficacy is determined at 30 seconds or 10 minutes post-treatment, and persistent is determined at that 12 hours or days following post-treatment. The pre-injection skin preparation, for pre-injection skin preparation, the skin site is an area that is commonly used for transepidermal injection or phlebotomy. This would be a dry skin site, the median cubital region. Recommended sample times are 30 seconds. This is just for going in and getting an injection. So quick disinfecting of the site and then an immediate sample after application. And that efficacy is determined at that, it just determines the immediate efficacy of that product. And we get into the hand studies. So ASTME 1174, this is for healthcare personal hand wash products, so water-aided products. This method determines the immediate and residual antimicrobial effects of a healthcare personal hand wash. This will use the glove juice sampling technique. Subjects refrain from using antimicrobials for at least a week before testing. This measures reductions of transient microbial skin flora. These are the microorganisms that we apply to the hands. Hands are artificially contaminated with 4.5 mils of a 24-hour broth culture of S. marcescens, ATCC 14756. E. coli can also be used, 11229, if the product, if you want to target healthcare or food service market, can alternatively be used. For any study that uses a transient microorganism, you need to have an antibiotic sensitivity profile on hand, and it's required for treatment of any kind of infection-related adverse events. So that's whichever physician is treating the subject that's having an event will have that sensitivity profile at hand, and it'll help them in treatment of that event. The S. marcescens is typically used for these studies because it produces a red pigment when grown at 25 degrees on soybean casein digest agar. It's really easy to use, really easy to count. For E. coli, a plating agar with a suitable indicator such as mug, which produces a fluorescent byproduct, is listed in the method. We have used that. We have also used McConkey agar, where the bug will grow up purplish pink, and it's easily identifiable. For this study, subjects will perform a series of hand contaminations and hand washes. Sampling performed after an initial contamination is for baseline. Then subjects will perform a single wash followed by a sample for the measurement of immediate antimicrobial efficacy. For measurement of residual antimicrobial efficacy, there is an alternative hand contamination and recovery schedule. Subjects will perform an additional 10 contamination and hand washes. A final sample is performed following that final hand wash. A residual antimicrobial efficacy is determined by comparing that reduction observed after that 10th wash to the first wash. After completion of testing and before exiting the study, subjects should perform a 1-minute rinse with alcohol, perform a 4-minute scrub with 4% CHG or chlorhexidine gluconate hand wash, and apply a topical antibiotic. And anytime you use a transient microorganism on a subject, they should do a decontamination procedure before they leave the laboratory. And here's the hand contamination recovery schedule in a nice, easy format. So you can see the subjects will go through a cleansing wash. That's just to remove any kind of bugs, dirt, oil that they may have picked up coming into the lab. This is followed by a baseline sample where you contaminate the hands and then sample immediately. After that, you do another cleansing wash. And then after that, then you apply the microorganism again and then do your first wash. That's for your immediate efficacy. And then you do a cleansing wash after that, and you start all over again. If you're doing a residual evaluation, you'll do another 10 applications. So washes 2 through 10, you'll do contamination prior, followed by the test formulation, use of the test formulation. And then after that 11th one, or the 10th post-wash one contamination and wash, you'll do a sample of the hand, and that's residual. So if a product has a residual or a buildup effect on the hands, you'll see that residual efficacy will be greater than the immediate efficacy. In the old 1994 tentative final monograph for healthcare personal hand wash, the reduction requirement after wash 1 was 2 logs, and then it was expected that that product would have an increase in reduction following similar contamination wash schedule at wash 10 of 3 logs. And after that, subjects will do a hand decontamination procedure next at the lab. E2755 is a method that was developed after the alcohol hand gel started becoming popular. This determines the immediate and residual antimicrobial effects of a healthcare personal hand rub formulation. This is intended for hand rubs, hygienic hand rubs, hand sanitizers, hand antiseptics, any kind of waterless hand disinfecting products, hand towelettes, anything where you would not use water. This employs a low volume liquid hand contamination procedure using a prepared high titer inoculum. Alcohol-based hand rubs are recommended for use on hands not visibly soiled. So hands visibly soiled would require a soap and water wash. This method simulates when the hands are not visibly soiled. So you have essentially a dry contamination. After you do that small volume, the hands are dry pretty quickly after that small volume application, and then you can apply the sanitizing product. This uses the glove juice sampling technique. This measures reductions of transient microbial skin flora. The subjects refrain from using antimicrobials for at least before testing. Again, the hands are artificially contaminated with a low volume. This is 0.2 mils or 200 microliters of a prepared culture vest marcescens. This marcescens is prepared in a broth that's vigorously aerated during incubation. That broth suspension is then concentrated using centrifugation, and the test inoculum that is prepared is usually 1 × 10⁻⁻⁻ CFUs per mil. So it's higher than what we would typically see in the E1174, which would be around 10⁹. Staph aureus can also be used in this study. That doesn't require centrifugation. It grows up very well during the vigorous aeration. Again, you're using a transient microorganism, so you should have an antibiotic sensitivity profile on file in case there are any adverse events that you'd be treated related to the microorganism. S. marcescens produces that red pigment when growing, so it's really easy to count. For Staph aureus, a plating auger with a suitable chromogenic indicator such as hardy chrome Staph aureus is recommended in the method. That media can sometimes be hard to find, so we've had some good success using a mannitol salts auger also, where the Staph aureus grows up nice golden color. You're contaminating the hands with a high enough concentration of the microorganism that it completely negates or covers up the any normal flora that you may get in those samples. Again, subject will perform a series of hand contamination and hand rubs. Sampling performed after initial contamination for baseline. And then there's an additional step in this one that's different from the 1174. This one, subjects will perform a single hand rub with a reference control, 60% isopropanol, followed by a sample. This reference control helps to control for inter-subject variability, inter-experimental variability, inter-laboratory variability. If you're running a lot of these studies, this helps you to standardize the data between those and help you to compare formulations over years or over multiple studies. It's an improved statistical comparison of test material using differences in log reductions between the reference control and the test material. So you use the reference control reduction minus the test material reduction, and then that value you use and compare that to your other studies. Subjects perform a single hand rub followed by a sample for measurement of immediate antimicrobial efficacy. That's after you do the reference control procedure. For measurement of residual antimicrobial efficacy, additional hand rubs are performed. So this one you perform 9 additional hand rubs. There's no hand contamination in this method prior to those 9 hand rubs. Prior to the 10th additional hand rub, subjects' hands are contaminated prior to that, and then a final sample is performed following that final hand rub. Residual antimicrobial efficacy is determined by comparing to the reduction observed after the first wash. So if the product has any kind of residual effect on the hands after those multiple applications, you'll see an increase in reduction after that final rub compared to your first wash or first rub. After completion of testing, again, before exiting the study, have the subjects perform a decontamination procedure. This is the hand contamination recovery schedule that's for this type of study. We have a cleansing wash. Now, after each one of these, since this is a dry procedure, there are some waits. So after the cleansing wash, there's a 5-minute wait before you proceed to your baseline procedure. Then after that baseline procedure, you do a cleansing wash, and you wait 5 minutes after that, and then you do a reference control procedure. Then after that, you do a cleansing wash, and then that one you wait for 20 minutes before you start the test product. So if subjects are going to be sitting around waiting quite a bit longer in this one than they do in the 1174. And then after that first application, they do another cleansing wash, and then they go into the applications 2 through 10, and then application 11, they'll do another contamination, then test material application for your residual efficacy. Then they do the decontamination before they leave the lab. For determining, there are 2 methods primarily designed for determining residual effectiveness. And first one is ASTM 2752. This is for residual effectiveness of antibacterial personal cleansing products. This determines the residual antimicrobial activity by inhibition or proliferation of bacteria on the skin after a contact time. This uses the cup scrub procedure. Subjects refrain from using antimicrobials for at least 2 weeks prior to the test day. This is performed using the volar surfaces of the inner forearm. One arm is treated with a test material, and the other arm is either treated with a non-antimicrobial vehicle product or no treatment. 3 applications are performed daily with 1 hour between application. Applications performed for 3 consecutive days, completing 9 total applications of product. You can do other application frequencies. We've done this after a single application. We've done this after multiple applications for 5 days. It's whatever you think you need to do for your product to evaluate its residual efficacy. Residual activity is assessed at times after that last treatment. So after you do that last treatment, 1 hour, 6 hours, 24 hours, or however long after that, you can do other times. 3 circular sites can be evaluated on each forearm. Use a sterile cylinder used for marking out those sites. A 4th site may also be used evaluating depending on the size of your cylinder and the size of your subject's forearms. On each one of those sites, 10 microliters of a microorganism is applied to the center of the circular site. The method recommends Staph aureus ATCC 27217 or E. coli ATCC 11229, or other appropriate bacteria cultures may be used. We've used a number of different microorganisms, fungi, viruses with this basic design. Culture concentration between 10 to the 8th and 10 to the 9th. So you're putting 10 microliters on there, you're exposing the site to 10 to the 6th, 10 to the 7th colony forming units. For plating samples, use appropriate plating media to aid in identifying and enumerating a contaminating microorganism. You're using Staph aureus and E. coli in this one, so you can use the Hardy Chrome or the mug, or you can alternately use the MSA, Manifold Salt Agar or McConkey Agar for a cheaper option. Again, have the antibiotic sensitivity profile of the microorganisms that you use in this study. to help treat with any adverse events that may occur. Inoculated sites are immediately occluded with an occlusive chamber and secured to the skin with an adhesive dressing. So here you have 2 sites that have already been covered and 1 site that's being contaminated and will be covered. We just use way boats with tape to occlude that site. Inoculated sites are sampled for surviving organisms after a predetermined period of exposure. Examples provided in the method are 30 plus or minus 5 minutes, 2 hours, 5 hours. We do not recommend longer than a day or 8 hours. 5 hours seems to be the max time that a subject will sit there. You can't leave let the subjects leave the facility once they have these way boats or the sites occluded on their arms. Again, I'm walking around with microorganisms on their forearms. So they have to sit in the lab and they will sit there for a period of time, but the longer the time period is, the more difficult it is to get people to volunteer for your study. You can contaminate those sites at any time after that last treatment. You can do 4 hours, 5 hours, 8 hours, 1 day, 2 days, 3 days after that last treatment. The longer you go after that last treatment, the less effective that product might be as the subjects are walking around doing whatever they want to do. You can try to restrict their activities during that time period, but the longer that time period is, the more difficult it is to evaluate that residual claim once you start going out multiple days. After each application, you disinfect each site after sampling with 70% ethanol. So if you're doing 32 hours and 5 hours, so that you do that 30-minute sample, disinfect the site. We just use a gauze pad soaked in 70% ethanol, wipe the site. After the last samples have been collected, you have the subjects wash both farms for at least 60 seconds with a CHG or 4% chlorhexidine gluconate hand wash, and then apply a small amount of antibiotic ointment over the sites. Residual antimicrobial efficacy is determined by comparing to the recoveries observed after exposure to the test sites treated with a test material to the sites treated with a non-antimicrobial vehicle product or no treatment. A relatively newer method that was developed was E3058. This is for residual kill activity of hand antiseptic formulations. This will determine the residual antimicrobial effects of skin antiseptics. Appropriate for water-aided or waterless products, the finger pads are used for the assessment. This uses a dry contamination procedure. So this is instead of 10 microliters of a wet inoculum applied to the site, this is instead will expose the site to a dry contamination procedure. Subjects will refrain from using antimicrobials for at least a week before testing. A minimum of 8 subjects is recommended per test material for this study. This measures reductions of transient microbial skin flora that are applied to the skin after application of the product. Staph aureus ATCC 6538 is used as a contaminating microorganism. You could use other microorganisms, but this method was designed for this Staph aureus. You might have to do some additional R&D to validate the contamination procedure if you use other microorganisms using this method. A broth culture is used to inoculate soybean casein digest agar plates and incubated for 18 to 24 hours at 35 degrees C. The colonies from those incubated plates are then suspended in soybean casein digest broth to create a contaminating culture of a concentration of approximately 8.9 to 9.0 log 10 CFUs per mil. You can determine that with aid of an absorbance meter. For plating samples, use appropriate plating media aid in identifying and enumerating the contaminating microorganism. You in Staph aureus, use the hardy chrome or the mannitol salts agar. Again, you're contaminating the skin, so you have an antibiotic sensitivity profile on hand for treatment of any adverse events. Now, for contaminating the hands, you have to prepare stainless steel discs. These are, there you have aluminum bars with stainless steel discs attached to them so that they don't move. On each disc, you put 10 microliters of a microorganism applied to the center. Use the same pipette for each application. Do not spread on that side, just put it right in the middle. You perform this in a laminar flow hood and allow those discs to dry for 1 hour. After they've dried for 1 hour, you transfer them to a relative humidity chamber set at 50 to 60% and also set at room temperature for 17 to 24 hours. When you're prior to using this to contaminate the finger pads, you remove this from the chamber within 5 minutes of contaminating the finger pads. For the subjects, they first perform a cleansing wash. They'll wait 5 minutes before applying the test materials. Then they'll perform a hand wash or hand rub with the test material. You can do 1 application, 2 applications, 3 applications, whatever's needed for your product to build up on those finger pads. They assess the residual activity 30 minutes after treatment. So after that, however many applications you're doing, 30 minutes after that is when you start it's recommended that you start 30 minutes after that last treatment. You can do it other times if you want, but this method was designed for 30 minutes. Contamination of the finger pad is performed by pressing the pads of a digit onto a contaminated disc for 2 seconds. So you've pulled those discs out, you have them set up on a counter. And then you have the subjects first will put their thumbs each thumb will be put onto a disc for contamination for 0 time. So they'll press it onto the disc. And then that thumb will be sampled immediately for baseline recovery. And then the remaining digits are similarly sampled. If you notice go back to here, you have 5 discs. So 1 for each digit. So you can do the thumb, and then the index finger, ring finger, pinky finger. Since you have multiple finger pads that are contaminated, you can look at different sample times for each finger pad, and each sample will be done in duplicate. You have right and left hand, so that'll help with your statistical analysis of the data afterwards. Okay, so you sample the finger pads by having the subjects firmly rub the pad in a circular motion for 30 seconds in the bottom of a petri dish containing 10 mils of sampling fluid. So this is similar to the European method, where you put the finger pads of all fingers into a petri dish. This one you just do the thumbs. So this one you're just sampling 1 digit. After that application, disinfect with 70% ethanol. So we use a gauze pad so we can control the application of that ethanol. We don't want that ethanol splashing onto the other fingers that have been contaminated. And then each of the other finger pads can be sampled at whatever time period you want to do. If you want to do 30 minutes after, hours after, the longer that time period is, the longer the subject's going to have to sit there. That's one thing to keep in mind when you design your study. After that last sample has been collected and before exiting the lab, such as perform a 1-minute rinse of alcohol, 1-minute scrub with a 4% CHE gluconate hand wash, and applied topical antibiotic. So typical, you're putting bugs on the hands or on the site, so you have to have the subjects disinfect those sites before they leave. Residual antimicrobial efficacy is determined by comparing to the recoveries observed after exposure on the finger pad to the time 0 your baseline recoveries. So whatever the recoveries you get on those other finger pads compared to that baseline, that'll allow you to determine what kind of residual effect your product had. Okay, I'll turn this back over to Mike or Daniel. Thank you all for your time, and I'll address any questions that you may have. If you want to ask me any questions offhand, you can definitely contact me at my email.
Great. Thank you, Chris, for all of your insights. We've learned some great information today, and you've given us a lot to think about. We do have some questions that came in while you spoke. And if you have any other questions, please feel free to submit those now. So if we don't get to your specific question, like Chris mentioned, you can happily address it offline as well. So let's go ahead and start with our first question here. All right, Chris. The first one is, what sample size is required for an efficacy evaluation? Chris, you may be on mute.
Yes, sorry, I was on mute. Yeah, that will depend on how you use the data, either for submission, publication, or screening. The FDA has requirements of a minimum of 100 subjects required per test arm if you're trying to get your product approved as a healthcare personal hand wash, hand rub, as a surgical hand scrub formulation, scrub or rub, or a preoperative skin prep. And that's minimum 100 subjects. You may need more if the data that you recover from your study is highly variable. The EAN methods have set sample sizes within their methods because they've standardized their analysis procedures, or how you analyze the data after you've collected it. And if you're going for Health Canada requires products achieve a significant reduction with 90% confidence and 80% power. So there are some statistical considerations to take into to concern yourself with when you're determining what sample size you need for a study, and that will depend on how you're going to use it. I mean, if you're just using this for a screening study or screening of multiple products, you can use a smaller sample size. If you're intending to do this for submission, we recommend you do a smaller, if you don't have no experience with your product, to where we can actually get some data on it, maybe even do a smaller pilot study first and then go into a larger study, just to confirm that minimum of 100 is what you would need, not, you wouldn't need more than that.
Great, thank you for that explanation, Chris. Here's our second question. What is the maximum time point I can test my residual disinfectant at?
I tried to comment on this earlier within each of the methods, but you can set that to whatever time point you may think give your product advantage in the market, or whatever you think your product can achieve. Again, we've looked at hours afterwards, days afterwards. The longer you go, the longer time period you go after, you should do, if you're going to look at days afterwards, you should really look at 3 time points total. You should look at an immediate, or like a within a day, and then look at the next day, like a middle time point and then an end time point. So you can kind of see if there's any kind of regression in your residual efficacy over that time period, or if there's positive regression, or if it increases over time, or if it stays the same.
Great, thanks, Chris. A couple more questions that we have here. If I have multiple versions of my product, meaning different fragrances, do I need to test each version?
You should. We always recommend that you test each formulation of your product. Even if you've reformulated your product, you should confirm that it still has the same efficacy as your previous formulation. Or if you're doing multiple versions of a product, you should confirm that they all perform the same, if they're all going to be used the same. So yes, we do recommend that you test. If you reformulate, you should test your new formulation.
Great, thank you for that answer. Another question here is, for an antiseptic product being sold in both a consumer and healthcare market, are 2 tests required by the FDA?
The FDA has requirements for the healthcare market. And if you're going to target both markets, you should first meet that healthcare market claim. And then that usually suffices for any other kind of consumer use you want to do with your products. The FDA they don't have requirements for the consumer market. They just require that if you're going to make claims on your product, that you have data that supports those claims on hand, and that those data are actually are valid and support whatever kind of claim you're making for your product.
All right, we have one more question here. Can you make residual antiseptic claims against specific viruses?
There are ASTM methods for viruses, and there's ASTM methods for fungi, but you can modify, especially those 2 residual antimicrobial methods that I talked about. the E2752 and the E3058. I mean, those are set up for different microorganisms, but are set up for the ones specified in the methods, but you can use different ones. For that 2752, we've used other fungi, you can use virus. You just need to make sure you have appropriate controls in place for processing, or the procedures that you're using to sample whatever organism you're using are appropriate for the organism, that you're able to recover, that you're able to use it safely, that the subjects aren't going to have any adverse experiences related to whatever microorganism that you're using. But we've modified these methods for a number of different viruses and fungi and bacteria. So yeah, you can use these methods to prove that that your product has a residual effect.
Great. Well, we thank you, Christopher, for that thorough and informative presentation and for all your great answers on these questions. I'd like to thank Chris, our presenter, for his insight, and thank you, the viewer, for attending this session. We hope that you found it to be a valuable experience. You can find this session archived on the Nelson Labs website under the On Demand's webinars section in the next couple of days. It will also be a part of the Sotera Health Academy and we invite you to register for that academy for free. We thank you, and we hope you have a great rest of your day.